Okay, so as Tanya said, our group is interested in gastrointestinal and pulmonary microbiomes. And more recently, we've become really interested in how environmental microbial exposures may influence human health and dictate the propensity for disease development. And so we work in two major disease areas: inflammatory bowel disease, which is really centered within the gut and local inflammation, but also in asthma and childhood asthma. And our belief is that the genesis of this disease occurs because of a dysbiotic gut microbiome. And we've set about trying to prove this with a series of studies that I'll tell you about today. And so childhood allergic asthma is really the poster child for a disease that is influenced by the environment. The dramatic increase in prevalence shown here in the red bar, or the red line in this plot, is in children over the last several decades. And you can see that it has really accelerated in prevalence over just a few short years. And so this suggests to us that while there are genetic risk factors for the disease, there's a strong environmental exposure component that is associated with disease development. And that's also supported by the fact that the highest prevalence of disease are in developed countries. We really don't see a whole lot of asthma and allergy in developing nations. The disease is centered around the Americas and Australia. And Graham Rook, a number of years ago now, wrote a fine review examining human microbiological history. And so as we moved from hunter-gatherer populations over 10,000 years ago to kind of medieval agriculturists, we really didn't change our interaction with the microbial environment that much. We still lived primarily in rural environments. We had a lot of contact with soil, with feces. We domesticated animals at that stage. And we really had not changed our microbial encounters that we've evolved with. But it's really with the second very large epidemiological transition as we've moved into the modern era, post-industrial stage, post-industrial revolution phase, that we've really dramatically altered our interaction with the environment, and I think in particular with microbial exposures in the environment. We now primarily live in cities. Two-thirds of the population, and particularly in developed nations, live in urban areas. And we have far less microbial exposure. We drink chlorinated water, we use antibiotics, we have far less animal exposure. And back in 1989, David Strachan and colleagues in the British Medical Journal proposed what's well known as the hygiene hypothesis. And this is the idea. It's evolved somewhat since its original proposal, but it's the idea that it's a lack of exposure to commensal microbes that really is underlying this allergic asthma disease prevalence that we have in Western nations. And it's really a shift in lifestyle that underlies the disease. And as you all know, we're superorganisms. We're a conglomerate of mammalian and microbial cells, and we're far outnumbered by our microbial colonists. And the majority of these microbes, or a large portion of them, are found in the gut microbiome. And they contribute to essential functions to host health. They help us metabolize indigestible carbohydrates that we ourselves don't have the enzymic capacity to degrade. They're involved in vitamin and hormone production, essential vitamins that we need, but we either consume or these organisms produce for us, like vitamin K. They're critical and essential for immune development, and we know that from germ-free mouse studies in which these animals, without commensal microbial colonization, are physiologically and immunologically aberrant. And the only way to overcome these aberrancies is by colonizing the mice with commensal organisms. We also know that specific organisms belonging to key groups like the Clostridia are engaged in dictating the tone of host immune status and can actually induce populations of immune cells that can quench pro-inflammatory responses. And we and others have shown that the microbes that reside on the mucosal surface, we've shown it in the upper airways, others have shown this in the gut, participate in what we can see as ancillary mucosal protection. They can compete with pathogenic organisms and prevent their outgrowth or invasion into the ecosystem, thus protecting the mucosal surface from infection. And as I said, we're really interested in childhood allergic asthma. And we were really intrigued by the fact that a large number of factors that are associated with the development of childhood allergic asthma in very early life are associated also with, or could conceivably be strong influences on the microbiome. Lack of maternal exposure to livestock or furred pets, and I will show you some data that shows how this may be influencing the microbiome. Formula feeding, we know, reduces commensal bifidobacteria in the gut microbiome of infants. Cesarean section delivery, some beautiful work by Maria Gloria Bella Dominguez has shown that C-section babies, which have a 30% higher risk of developing allergic disease and asthma, have a very distinct pioneering colonization pattern of microbes in the mouth, in the gut, and on the skin when they're born by C-section. Vaginally born children start life with Lactobacillus and Snethia organisms, and we think ecologically that really influences the development of the microbiome at these various niches in the human body, and that's what dictates the propensity to develop disease at a much higher rate in these children. We certainly know from David Relman's work that antibiotic use has a profound effect on the microbiome, and we know that mothers who are administered antimicrobials during pregnancy, their children are at a higher risk of developing disease. And also the children who, again, receive antimicrobials in very early life, again, have a much higher odds ratio of developing allergic asthma. So it occurred to us that really the risk factors, or many of the risk factors for developing childhood allergic asthma, are really critically centered around the prenatal and the neonatal phase of life. And this is supported by 2 independent studies of large numbers of children who showed that early life enrichment of Clostridium difficile, or E. coli, in the fecal material of children as young as 3 weeks of age was associated with those children going on at a very high rate to develop allergic asthma in childhood. So what happens to the gut microbiome in very early life? And I'd say that this happens, we suspect, across all mucosal surfaces, but the gut is the site that we've studied the most intensively. And this is from a study that we have ongoing. These are simply time points in the first year of life of 25 individual infants, where we collected stool sample at birth, so meconium, or their first bowel movement, 1, 3, 6, 9, and 12 months of age. We profiled their bacterial communities present, and we simply calculated Fate's phylogenetic diversity. And what you can see is across all children develop this phylogenetic diversity, or this expansion of phylogenetic diversity over the first year of life, irrespective of the breadth of diversity that was present in the earliest stool sample that we collected. And so it occurred to us, well, if this is the breadth of diversity that children are born with, and they end up somewhere around here at the end of the first year of life, where does that microbial exposure come from that allows these children to accumulate microbes into the developing gut microbiome? And a study by Calif and colleagues in 2001 examined how much time in Western nations we spend indoors. And it's actually quite staggering. It's almost 90% of our time is spent indoors, with a very large proportion of that in our residences, almost 70%. And so collectively, we brought all of these observations together to propose a causal pathway for childhood allergic asthma development, in that the local microbial environment in the home of a child maybe influences their microbiome development in a manner that dictates a host immune response and disease susceptibility. And so the first thing we asked, if this hypothesis was true, was whether the environmental microbial exposure in the homes of children with dichotomous risk for allergic disease development was distinct. And so our colleagues at Henry Ford Hospital, Christine Cole-Johnson and Dennis Ownby, had made a really wonderful observation over 10 years ago now, published in JAMA, where they noted that infants who were exposed in very early life to dogs and to a lesser extent cats were protected against childhood allergic disease development. And so we postulated that this might be due to a distinct microbial exposure in these homes where children were at dichotomous risk for disease development. So we took house dust, collected in a 1 meter square quadrant from homes with dogs, cats, and no pets present. And we used a phylogenetic microarray to just give us a broad view of the bacterial community that was present and compare it across these distinct environments. And when we looked at alpha diversity metrics such as community richness, or the number of types of bacteria that we detected, their distribution within the community, or evenness, and the diversity within the communities, what we could see is that consistently the homes with no pets present were significantly reduced in these metrics compared to dogs, and to a lesser extent, cats. So homes in which there were no pets present, which confer a higher risk for developing allergic disease and asthma, are microbially deficient. And I won't give you a long list of Latin names that makes no sense really to anybody, but what we can say is that the majority of bacteria that we found significantly enriched in these protective homes that had dogs present had already been described in the human gut microbiome. So we were really intrigued by this observation. We've actually subsequently confirmed it in an inner city cohort of children, over 100 children, where we know their clinical outcomes of allergic disease and asthma at age 3, and shown the exact same trend, that children who go on to develop disease lack bacterial exposure in the very earliest stages of life when their gut microbiome and other microbiomes at other niches are developing. So we have good evidence that there is a distinct microbial exposure associated with disease development or protection against disease development. But what about these other parts of our proposed causal pathway? Well, to examine this, we partnered with Nick Lukacs at the University of Michigan and Tina DeMoore, and this was driven in my lab by Kei Fujimura, a very talented staff scientist. And what we did was we took house dust from the extremes of microbial exposure, from homes with dogs present or no pets present. And our institutional review board won't let us feed babies dust. Don't know why. So we turned to mice. We thought that would be a better model to go with to try and prove our hypothesis. And so we set up a kind of an unconventional model, but nonetheless one that was trying to recapitulate the environmental exposures that children would encounter in these homes. So we fed our mice a daily oral gavage of these distinct dusts, and we had some control animals as well, for 7 days, with the idea that we might shift the gut microbiome. And then over the subsequent 2 weeks, we challenged their airways. We sensitized them to cockroach allergen challenge. So we literally intratracheally induced sensitization in the airways of these animals. And during this period, we continued that exposure to these distinct house dusts, but at a less frequent interval, twice per week. And what we found was that the animals that were orally gavaged with the dog-associated house dust had significantly reduced hallmark cytokine responses of allergic response in their airways. IL-4 and IL-13 are the two primary cytokines expressed by Th2 cells, which are the characteristic response to an allergic response. They also had significantly less mucin in their airways, as characterized by significantly reduced GOB5 expression. And really, I thought the histology was the real clincher. These are control animals that we did not do anything to, no oral supplementation, no cockroach allergen challenge. This is their airways. Their air spaces are pristine. These are animals that are simply exposed to cockroach allergen. You can now see that the airspace is occluded with pink staining mucin, and these airspaces, it's hard to see, but there's a lot of nucleated cells around them. They're highly inflamed. These are the animals with the no pet dust exposure, and you can see again a lot of mucin hypersecretion, and there's a lot of inflammation in these airways, consistent with what we're seeing in the molecular signature. And these are the animals who were fed dog-associated house dust, and their airways by histology look pristine. But remember that the hypothesis was that it's changes in the gut microbiome that allow for protection of the mucosal site in the airways. And so we examined the cecal contents, the bacterial community and the cecal contents of these animals. Control animals shown in here in green, animals fed no pet-associated dust in red and dog-associated dust in blue. And this is a principal components analysis. The way to interpret this is each one spot represents a bacterial community, and its proximity to another spot tells you how similar these communities are. And this was based on the Canberra distance for those who are interested. And what we could see is that feeding dust really does shift the gut microbiome composition of these animals, and that we could explain quite a large amount of variation in these communities based on dust exposure. But what we were really interested in was who specifically was enriched in the gut microbiome of animals who were protected. And what we found was that there was about 100 taxa that discriminated protected and unprotected animals. And one of these was the fourth top-ranked finding, was Lactobacillus johnsoni. And we became very interested in this because this is an organism that becomes highly enriched in the mother's vaginal tract just before she gives birth. suggesting to us that it could be a strong contender for an organism that's inherited by vaginally born infants and that it may strongly influence gut microbiome composition. So we went back to some good old traditional isolation techniques for Lactobacillus. We took another group of mice, we fed them more dog-associated house dust, and we isolated the dominant Lactobacillus present. We took 4 mice, 5 isolates from each of the 4 mice, and sequenced the full-length 16S. All of them were Lactobacillus johnsoni. So our molecular tools had accurately identified the dominant and most enriched Lactobacillus in these protected animals. And then we repeated the same model again, but this time we just used that single Lactobacillus species to ask whether it could confer protection that we'd seen previously with dog-associated house dust. And what we found was that it was a resounding yes. Again, this is cockroach allergen challenge of the airways, and we use this metric, it's airway it's how quickly the airways respond to a challenge. And we can see that they are significantly reduced in their responsiveness or their allergic status when animals are fed Lactobacillus johnsoni. Likewise, again, these Th2 allergic responses in the airways, IL-4, IL-5, and IL-13, are all significantly reduced when we simply feed the animals Lactobacillus johnsoni, and again, the airway histology looks quite improved compared to the control animals, though I would say not as pristine as the animals who received the dog-associated house dust. And we've indications from other studies that it requires a consortium of organisms, all of which are not necessarily found in a wild-type mouse, and some of which may be environmentally sourced for this full protective effect. The other thing that was of interest to us was that one of the other ways to develop asthma is to have a hallmark is to have a very severe response to a viral infection in very early life that requires hospitalization. And so there's a number of viruses that are considered asthmogenic, such as respiratory syncytial virus. And so we next asked the question whether Lactobacillus johnsoni supplementation could protect against viral respiratory infection of the airways, and whether we required a live organism to confer this protective effect. And again, this is the experiment. These are the data looking at bronchial hyperresponsiveness. Only the animals that received viable Lactobacillus johnsoni have less responsive or allergic airways. Those who received an equal number of heat-killed organisms do not have this significant reduction in bronchial hyperresponsiveness. And again, in the airways, in response to respiratory syncytial virus, we see a quenching or a significant reduction in adaptive immune responses associated with the Th2 response, here IL-4, IL-5, and IL-13, and the Th17 response, which is mounted against RSV infection and is characterized by IL-17. What was interesting to us is that we do not see a significant reduction in interferon gamma responses, and these are necessary for a mammalian system to clear a viral infection. So it seems like the mouse can still clear the virus, but does not mount the adaptive immune response that causes the ensuing pathology associated with this viral infection. And so it suggests to us that the presence of these organisms is modulating adaptive immune responses in a manner that prevents allergic response. The other thing we asked is whether the Lactobacillus johnsoni, as we suspected, had the capacity to shift the gut microbiome by any large measure with respect to composition. And again, this is another principal components analysis, and I want to draw your attention to these animals here who received Lactobacillus johnsoni, who all cluster together and are significantly distant from the control animals who received no supplementation, suggesting that these are compositionally distinct communities in the animals who received this single species. And collectively, this suggested to us that alterations in gut microbiome composition is associated with airway protection against allergen challenge and viral respiratory infection, and that a live organism is necessary, suggesting to us that there may be microbial activities or perhaps even metabolic activities that are associated with this protective effect. And so to address this, we've begun to look at a birth cohort of stool samples and paired house dust samples, which I won't talk about today, and I'll just focus on the stool samples. This is an amazing cohort of samples collected by Christine Cole-Johnson and Dennis Ownby in their Wayne County Health Environment Allergy and Asthma Longitudinal Study, or WHEEL study. It's centered around Detroit. They started collecting samples from these children in 2003, and they're still collecting the samples. We're in fact bringing the children back in now for a 10-year assessment of their allergic and asthma status, as well as a number of other diseases such as diabetes and obesity that we're examining in this cohort. They're from a socioeconomically and racially diverse community, about 50% are minority. We have 300 infant stool samples, and they range in age from birth to 11 months. It's a population-based cohort, so it's not biased in any way. We can ask any question of this cohort, which is really very powerful. And we've examined the gut microbiome of these 300 initial infant stool samples using the Illumina MiSeq platform and sequencing the V4 region of the 16S gene. And we have defined allergy or atopy at age 2 using a very new and very powerful approach called latent class analysis. So much as we are kind of agnostic in our allowing microbial communities to cluster together, we're taking the same approach with clinical data to classify children as allergic or not. So the way we look at allergy is we skin prick test. So we expose children to allergens and then look at their immune response or the wheal size that occurs around that allergen. And if we look at this data agnostically, what we find is that there's a group of children that we call predominantly multisensitized, and in that they're allergic to many of these allergens that we expose them to. And this group have a significantly higher risk of developing asthma at age 6 and subsequently at age 10. So this is how we divided our cohort into allergic children and non-allergic children who had low or no sensitization to any of these aeroallergens, such as dog, cat, and cockroach, etc. And so for the first thing we did, we asked, well, does the gut microbiome of these children follow the central tenets that we know to be true in that it changes with age over the first year of life? And indeed it does. This is a cross-sectional study, so these are independent samples from almost 300 children. And what we find is that 5.6% of the variation in observed community composition is explained by age. But there's a range of other factors that are explaining this variation, including mode of birth, which again, there's evidence for in the literature, and breastfeeding, which again, we know influences the gut microbiome. But it suggests to us the factors that we know to influence gut microbiome development and composition in the first year of life hold out and are validated in our cohort. But remember, many of the risk factors that have been identified as associated with childhood allergic asthma development are centered around the very earliest stage of life. So we parsed our data into children who were younger than 6 months of age and used a Dirichlet multinomial mixture model to statistically define infant subpopulations based on microbiome composition, microbiotype or enterotype or whatever you would like to call it. And what we found is that the model was best satisfied by 3 distinct types of microbiome in the gut of these children. One was dominated by Bifidobacteriaceae, one by Enterobacteriaceae, and the third was co-dominated by both of these families. And the co-dominated enterotype was really interesting to us. It was only a small number of children, but these lacked key commensals such as Akkermansia mucinophila, Faecalibacterium prownitzii, and Lactobacillus, Lactobacillus ruminus. And several of these and others or other organisms that were conspicuously absent from these children are known immunomodulators and have the capacity to induce T regulatory cells which dampen those pro-inflammatory responses associated with allergy. So we next ask, is there any clinical correlate with these different types of gut microbiome that occur in children over the first 6 months of life? And there's a significant relationship with the risk of allergic disease or atopy development and which type of gut microbiome is present in these children in the first 6 months of life. And the co-dominated community that lacks many of these commensal bacteria is the one that confers the highest risk for allergic disease development. And I should have said at the outset, many children who develop severe allergies go on to develop asthma. It's kind of called the allergic march. It's not all allergic children develop asthma, but many allergic children who have more severe disease do. So it looks like we're seeing that there is a very early life gut microbiome composition associated with allergic disease development in our cohort. And we wanted to move beyond descriptive community composition towards function. And we asked the question, are compositionally distinct neonatal gut enterotypes functionally distinct? And we kind of cut to the chase. We didn't do metagenomics and we didn't do transcriptomics. We went directly to metabolomics. And we did this because we were intrigued by the observation that the live lactobacillus was necessary to confer the protective effect in mice, suggesting to us that it may be actually metabolic activities in part of these distinct microbial communities that is associated with this distinct risk for disease development. So we performed global LC mass spec on 30 samples, 10 from each enterotype. And what we could see is we hit close to significance, but there are differences metabolically between these 3 infant enterotypes. And we reasoned that there's probably metabolites in common to all infant gut luminal contents, and that maybe is what's masking some of the difference here. But we asked specifically what characterizes the children who have that high-risk co-dominated community. And what we find is that they are significantly altered in their capacity to metabolize carbohydrates. Specifically, di and oligosaccharide metabolism is highly altered in these children, and their capacity to metabolize lipids or fatty acids. And I'm showing you just 2 data points among several that are of key interest to us. What we see is that the children with the highest risk of atopy development are already enriched in very early life for common pro-inflammatory lipid mediators, 12,13-dihome and 13,9-HODES, which are associated with severe asthma development in adulthood and in childhood when it's established. So collectively, what we're starting to understand by piecing together compositional analysis with functional analysis, mouse studies with human studies, is that it appears that the foundation for atopy development in childhood is associated with a compositionally distinct and dysfunctional neonatal gut microbiome, and that the gut microbiome is in turn influenced by the local environmental microbial exposures of that child. And so our current developing model for atopy development is that the gut microbiome is influenced by factors such as breastfeeding and mode of delivery, which are associated with distinct community compositions in very early life. That this leads to shaping distinct enterotypes in children in the very formative weeks of life. And that these distinct microbial enterotypes are functionally distinct and are metabolically very different from one another. And that they produce metabolites, especially the co-dominated community that's associated with higher risk, that discretely influence the adaptive immune response, particularly the T-regulatory immune compartment, which we've evidence for in some of our earlier studies. And that this distinct immune priming of the host immune response in very early life is what ultimately leads to downstream atopy or non-atopic development in these children. And so I'd like to, obviously this is a huge amount of work from a large number of collaborators, and not least Homer Boucher, Kei Fujimura, and Doug Fadrush, who have been just really done some amazing work at UCSF with us. And Chris Johnson, Ed Zarate, and Al Levin at Henry Ford Hospital, who've kindly given us access to an amazing birth cohort that we have been examining. And then Dennis Ownby, who's also been involved in the Henry Ford Hospital cohort, but has since moved to Georgia Health Sciences University. And Nick Lukacs and Tina DeMoore at the University of Michigan, who have helped us with some of our murine studies to really get down to the molecular mechanism as to how these manipulations of the gut microbiome may modulate immune responses at a remote mucosal site. And then finally, I would like to thank our funders, the NIAID and the Fund for Henry Ford Hospital. And I'm happy to take any questions.
We have time for a few questions, yeah.
Thanks for a great presentation. My question would be, it seems like the dominant bacteria between these 2 groups is Lactobacillus, and you also mentioned the Bifidobacteria. Actually, that's are the 2 main species that's in yogurt. So the probiotics, now in the supplement, says there is no clinical proven thing that is beneficial. In explaining this difference, have you analyze the diet, the diet habit of the mom and also the children?
We haven't looked at the mom. The next round of our studies will be examining prenatal exposures because there's a great dearth of information in that field and how prenatal diet and microbial exposures of the mother may influence the immune status of the child at birth. So we can look at cord blood, for example. We do have dietary information for the children. I mean, it's very easy, and that's one of the reasons we looked at pre-6 months of age, because they're either breastfed, formula-fed, or a mix of the two. And so what we see is that one of the two factors that have the strongest influence on gut microbiome composition are cesarean section and breastfeeding. So diet plays a very strong role in this. But what we think is that diet supports some of these organisms, such as bifidobacteria, and then the bifidobacteria, together with the diet, dictates the ecosystem conditions that permits environmental organisms to co-colonize with that dominant organism and its kind of guild of organisms that co-colonize with it. But it's certainly something that we're looking into, yes.
Yeah, maybe a survey about their diet habits if they consume extra yogurt. Compared with the other group
I'm always wary about saying yogurt varies tremendously across brands and types. I mean, there's brands out there that are just pure sugar, which is not going to do good things for your gut microbiome. And there are others that house live organisms that may be beneficial, though I'm not sure that the numbers found in yogurts, even live yogurts, are sufficient to shift the microbiome. There was a study by Jeff Gordon's group showing that consumption of fermented milk didn't really do much to change the microbiome, though I would add that the caveat with that study was a very shallow 16S profiling. It was 1,000 reads per sample. So I would say it doesn't change the dominant organisms of the gut microbiome, but that's not to say it may have some influence on the lower abundant organisms. But really, quite honestly, the studies have not been done yet. But I think diet is going to emerge as one of the largest adjuvants to therapeutics that we commonly use, that we currently use, and that everybody's health is in their own hands, essentially, through dietary interventions.
Thanks a lot.
I found the results with the dog exposure be very interesting. So do you have an idea where those microbes are in or on the dog, and could you substitute the dog with, say, a really dirty uncle or something? I mean...
Are you really going to make me say it? We think it's actually the dog's stool, because so many of the organisms that were enriched were actually gut organisms. We think that maybe the dogs are outside and being exposed to environmental organisms and then passaging them through the gut. But mostly what we saw were gut-associated organisms. That's not to say that saliva might not have some organisms that could be beneficial. I know that there's a study about to start in Arizona to look exactly at that, to look at saliva of dogs, because they hypothesize that dogs lick everything, so that perhaps that's where the beneficial organisms are. I think some of this is simply exposure to commensals as well, and it's just another, children, birth order is another factor that plays into risk for allergic disease. So if you've got 7 kids ahead of you, you have a really low risk of developing allergies. So, we say 1 dog is as good as 2 brothers.
I'm wondering, how do you have any data showing how quickly the effect of microbial exposure on the immune sensitivity changes with age? So, like these mice experiments that you did, the data is beautiful when you look at infant effects, but then later in the life, are we doomed if the disease develops?
We haven't done them, but others have done those studies, and there's actually data as well. They've done those studies in mice where the presence of specific organisms in early life are protective, but finding those, adding those organisms later in life when the microbiome has developed is less protective. We did our studies when the mice were 6 weeks of age because we felt like they were still developing, and that would be a good period where they were still somewhat plastic. There's also evidence from humans that finding Moraxella and Streptococcus, and I can't remember the third organism, in the nasopharyngeal or the oropharyngeal microbiome in the first few weeks of life is a high risk for allergic disease development. But finding those same organisms at 12 weeks of age is not a risk. So context matters, and that's what we're really after, is understanding how these organisms work as units together, or even subsets of organisms within these large consortia, act as guilds of organisms that interact to produce these protective metabolites that we're finding in some of our children. And we are starting to see, we've kind of worked backwards from the metabolic profile and found that the metabolites that are associated with protection are really quite interesting, and some of them are known to be immunomodulatory fatty acids and things. And we're working back with the metagenomics and predictive metagenomics and 16S data to figure out, are there organisms enriched in those environments that have the capacity to produce those lipids? And there are. So how we're developing our therapeutic consortia is working from a functional differential between health and disease and working back towards the organisms that produce those metabolic products that confer the beneficial effect. And we find them in the healthy kids.
So I have a question.
Yes.
Why dogs and the cats don't have the same effect? Is that different gut microbiota, or
That
is
may
the
be
type
it.
of lactobacillus?
What was really interesting to us was that the original study showed dogs and to a lesser extent cats are protective. And what our data showed, just from a very gross overview, is that dogs increase the diversity to the greatest extent. Cats, there's a smaller diversity increase compared to no pets. So I don't know whether it's a different gut microbiome, whether they're just smaller animals and bring in fewer organisms from the environment. One of the things I didn't show with that study is that the amount of diversity was related to the behavior of the animal and whether they were indoor, outdoor animals. And we think cats may be more likely to be kept indoors exclusively. And if that's the case, then they would not have that environmental exposure and the capacity to introduce environmental organisms into the home of those children.
You think? Oh. You're showing that the Lactobacillus has a huge effect?
Yes,
that
yeah,
particular species is not present in the cats, or the cats have a different type of that species?
We haven't looked for that. That's a good point. We haven't looked. We know that that Lactobacillus, so this is the interesting wrinkle with this, that Lactobacillus is not highly enriched in either the dog or the cat environmental samples. So what we think is that exposure to those environmental organisms allows for enrichment of a native Lactobacillus in the gut microbiome. I don't necessarily think it's coming from the environment itself, though I've yet to be proven wrong.
Susan, do you think cats could just be cleaner in general as an animal?
Possibly. And, I don't like dogs, so yeah.
I like cats.
Yeah, cats are great. Cats are great.
Okay, I think we had one more question in the back. No? Okay, so I think it's time for a break. Let's give her a hand.